Spatial transcriptomics is rewriting the early biology of lung cancer. Today we're looking at one of the most detailed maps ever created of how lung adenocarcinoma begins, from the first transitional alveolar cells to inflammatory niches to the earliest detectable clones. If you're studying lung cancer, tumour microenvironments, epithelial plasticity, this paper is a landmark, genuinely. Let's walk through what it reveals about how LUAD really starts. Welcome back to Further Reading. I'm Dr. Susan Heavey, Associate Professor at UCL, and today I'm going to walk you through this paper, panel by panel, so you can really see how each figure builds the story of early lung adenocarcinoma. It's a longer video than usual, and that's because this is a truly landmark study, and I want to give it the space that it really deserves. So grab a cuppa, relax. This video is especially useful if you're working on a dissertation in lung cancer. Maybe for your PhD or postdoc, you might be studying epithelial plasticity or trying to understand how spatial transcriptomics reveals biology that bulk and single cell methods simply can't capture. I'll be honest, it's a bit more of an advanced video. If it's all going over your head, you might want to skip ahead of the panel by panel to the Spatial Bio 101 section. Okay, so figure 1 begins with the overall study design. Here, the authors map the entire pathological continuum of lung adenocarcinoma development, from normal lung through atypical adenomatous hyperplasia and adenocarcinoma in situ, to minimally invasive adenocarcinoma and fully invasive LUAD. They use a genuinely multimodal approach. So we're talking Visium spatial transcriptomics to build whole tissue spatial maps, single nucleus RNA-seq from matched blocks to define cell states, Xenium single cell spatial transcriptomics to validate these states at cellular resolution, and even whole exome sequencing to infer copy number changes and reconstruct clonal evolution. Altogether, this pipeline generates one of the most comprehensive spatial atlases of early lung adenocarcinoma produced to date. And as a PI with limited funding, I gotta say, it looks like it cost an arm and a leg. Okay, so figure 1b shows the breakdown of more than 486,000 spatial spots grouped into epithelial and non-epithelial compartments. The key point here is how rigorously pathologists annotated every spot. We've got normal alveoli, precursor lesions, invasive tumour regions, lymphoid aggregates, stromal areas, vascular structures. It's all integrated. This careful annotation is what allows the latter analysis to link spatial gene expression directly back to histology. And again, I've got to say, as a PI, this is not just expensive, but extremely time-consuming work requiring really experienced and wise pathologists to give up an awful lot of their time for research. So again, full marks, round of applause. In figure 1c, the authors embed epithelial spots from precursor and invasive lesions into a joint UMAP. You can already see stage separation emerging. AAS and AIS cluster closely together, reflecting their early AT2-like gene expression. LUAD spots show much more heterogeneity and diverge widely across the map. The feature plots highlight this biology. Early lesions retain SFTPC consistent with AT2 identity, while invasive LUAD gains markers like KRT8, CCAM5, and mucin genes associated with dedifferentiation and tumour progression. Figure 1d zooms out across all 486,519 spots and shows a clean gradient of gene expression across histological states. Normal regions express AT1 and AT2 markers like AGER and PGC. Precursor lesions retain alveolar programs but begin to upregulate stress and transitional signatures. Invasive LUAD shows high CCAM5, EGFR, MUC5AC, TFF3, classical tumour-associated programs with increased plasticity. This heat map really illustrates the continuum the paper emphasizes, not a stepwise jump, but a progressive drift from alveolar identity towards tumour-associated metaprograms. The final panel, figure 1e, overlays marker genes directly onto H&E images at superpixel resolution. This is where spatial modality shines. You can see SFTPC clearly marking normal alveolar regions, AGER marking AT1 cells, CCAM5 highlighting tumour clusters, and MS4A1 identifying lymphoid aggregates. This panel makes spatial structure of the lesions visible, the alignment of molecular identity with architecture, and sets the stage for all the higher resolution niche and clonal analyses later in the paper. So overall, Figure 1 establishes the spatial landscape, which cells live where, how early and invasive lesions differ, and how alveolar programs shift across the pathological spectrum. Now that the spatial map is in place, Figure 2 moves into one of the most technically impressive components of the paper, reconstructing clonal evolution using spatial copy number inference. Figure 2 is another thing of beauty. This is where the paper really stretches beyond descriptive spatial maps and moves into evolutionary reconstruction. This is one of the most exciting parts of the study, using spatial transcriptomics to infer copy number alterations and rebuild the clonal trajectories that link precursor lesions to invasive lung adenocarcinoma. So panel A outlines the overall strategy. The authors used spatial infer CNV to infer copy number alterations directly from Visium spatial transcriptomics spots. Nice. They then integrate these spatial CNAs with matched snRNA-seq and whole exome sequencing to validate the signal and ensure the inferred clones are biologically meaningful. Finally, they use these clone calls to reconstruct patient-specific phylogenetic trees, layering each clone back onto the tissue in physical space. Panel B summarizes the evolutionary patterns they identify across patients. There are two main categories, pattern 1 and pattern 2. Pattern 1, where precursor lesions and invasive LUAD share at least one ancestral clone, but invasive regions acquire additional subclones as they progress. And then panel 2, where precursor lesions and LUAD share no detectable clones at all, suggesting independent evolutionary paths or very early divergence. This immediately highlights how variable early LUAD evolution is across patients. Panel C shows representative examples. On the left, for each patient, you'll see the phylogenetic tree derived from spatial CNV data. In the middle are the CNA heat maps, showing gains and losses across the genome for each clone. And on the right, each clone is mapped back into its anatomical location, alongside the pathology annotations, pseudotime ordering, and cytotrace differentiation scores. Let's focus on two examples. Firstly, pattern 1A. Here, clones present in the precursor lesion reappear in the invasive LUAD. The invasive tumour then branches further, acquiring additional subclones. This suggests a more linear progression, early precursor to shared clone to expanding invasive branches. Moving on to pattern 2, in this example, the precursor lesions harbour completely distinct clones that never appear in the paired LUAD. The invasive tumour is dominated by entirely separate subclones. What's fascinating is that in both patterns, the earliest identifiable clones often map to the reactive pneumocyte population, these KRT8 high alveolar intermediates. They appear to act as the earliest spatial foothold of clonal expansion. Panel D compares driver mutations across the two patterns. Pattern 1B, where precursors in LUAD share some but not all clones, shows the highest frequency of KRAS, EGFR, and MET mutations. Interestingly, KRAS mutations are absent in pattern 1A. This mutation stage association hints that genomic background shapes whether precursors contribute directly to invasive cancer or diverge early. These distinctions are important for understanding patient-to-patient variability in progression risk. So overall, Figure 2 shows that spatially reconstructed CNAs can reveal the hidden evolutionary relationships between early lung lesions and invasive tumours. In many patients, alveolar progenitors carry the earliest clones. In others, precursor lesions evolve independently from the eventual tumour. With the clonal and evolutionary framework established, Figure 3 turns to cell states and identifies the transitional alveolar intermediate population that links these clones to early LUAD biology. Figure 3 is one of the most exciting parts of the entire paper, although if I'm honest, that doesn't really say a whole lot because I personally at least find this whole paper pretty exciting. But this is where the authors trace the identity of the earliest epithelial precursors, the transitional alveolar cells that sit between normal alveoli and tumour. This figure shows, beautifully and convincingly, that these KRT8 alveolar intermediates are the epithelial cell state that emerges before recognisable precursor lesions. Panel 3A shows the single nucleus RNA-seq map of epithelial cells across all samples. You can clearly see the canonical alveolar lineages, AT1 and AT2, along with ciliated, club, basal, and tumour clusters. But the key feature is the distinct island sitting between AT2 and tumour, the KRT8 high alveolar intermediate cells, or KACs. Panel 3B colours each cell by lesion type, and straight away you can see the KAC clusters sit almost entirely within pre-invasive lesions. These cells are transcriptionally intermediate between AT2 and tumour, but spatially and biologically, they appear at the very earliest stages, that is before AIS, before MIA, and before LUAD. This strongly suggests that KACs represent the earliest epithelial state in LUAD evolution. Panels 3C and 3D quantify this transitional identity. In the pseudotime analysis, AT2 cells sit at the start, invasive tumour cells at the end, and KACs lie directly along the path between them. Cytotrace, which reflects developmental potential, shows the same pattern. KACs sit at a point of decreasing differentiation but increasing plasticity. These transitional scores support the idea that KACs are not random intermediates, they are a directional step on the route from alveoli to tumour. Panels 3E and 3F introduce the metaprogram analysis. Using non-negative matrix factorization, the authors derive 9 epithelial transcriptional programs, capturing AT1 identity, AT2 identity, stress and inflammatory signatures, tumour-associated programs, and more. KACs show a very distinctive MP profile. Reduced AT2 program, reduced AT1 program, but high expression of the tumour-associated MP6 and an inflammatory stress-related MP. In other words, KACs already share transcriptional programs with tumour cells, even while spatially appearing in regions that still look like precursor lesions. Panel 3G is a really important sanity check and really elevates the validation here, in my opinion. It cross-references these metaprograms with independent pan-cancer single-cell atlases. The correlation is striking. The same tumour-associated and stress-associated programs recur across cancers, and KACs fall directly into these conserved states. This anchors the biology. KACs aren't lung-specific artifacts. They sit within a universal spectrum of transitional epithelial states observed across many tumour types. Panel 3H shows how these programs evolve across pathology. AT2 and AT1 metaprograms drop sharply from normal to AAS, AIS, and LUAD. MP6, the tumour KAC program, increases progressively even in early lesions. This progression mirrors the morphological continuum and shows the transcriptional drift begins far earlier than histological invasion. Panel 3I overlays the metaprograms back onto the spatial transcriptomic maps. Non-invasive regions show predominantly alveolar programs, MP2 and MP7, whereas invasive regions show high MP6. What's powerful here is that the precursor lesions always show a mixed signature, alveolar on one side, KAC-like on the other. This highlights the spatial heterogeneity within early lesions before you ever see invasion. Panels 3J, which is a box and whisker plot, and 3K, which I'm showing here, take this even further. Using high-resolution I-star imputation, authors show that reactive pneumocytes, the morphological correlates of KAC in situ, cluster tightly around the edges of precursor and invasive lesions. These RP2 regions express a mixture of AT2 identity and tumour-associated programs. They look like the transitional zone where early clones expand. Panel 3L compares metaprogram abundance across normal alveoli, reactive pneumocytes, and lesions. RP2 show a hybrid identity, partly alveolar, partly tumour-associated, matching the transcriptional profile of KACs in snRNA-seq. Functionally, this positions reactive pneumocytes as a spatially defined precursor niche. Panels 3M and 3N integrate the clonal data from Figure 2. The earliest clones, the ancestral branches in a patient's tumour phylogeny, are disproportionately found in reactive pneumocytes. In some cases, these RP2-associated clones are shared across normal tissue, precursor lesions, and LUAD. In others, they're specific to precursor or invasive branches. Either way, the message is consistent. The earliest detectable clonal expansions occur not within obvious tumour regions, but within these transitional alveolar niches. Figure 4 goes on to take the cell state story from Figure 3 and adds the microenvironment. This is where the paper shows that the earliest alveolar progenitors don't sit in isolation. They live inside highly specific pro-inflammatory niches dominated by IL-1 beta producing macrophages. Panel 4A shows that KACs are strongly enriched for inflammatory pathways. Compared to other epithelial subsets, they upregulate NF-kappa B related programs, stress responses, and cytokine signaling. This already hints at that inflammation is shaping their behavior. Panel 4B maps metaprograms back to pathology. Precursor lesions and reactive pneumocytes correlate with a precursor MP. These same regions also correlate with a myeloid MP, particularly macrophage-associated genes. This aligns the transitional epithelial state with a corresponding inflammatory environment. Panels 4C and 4D highlight the key ligand-receptor pair. IL-1 beta from macrophages interacting with IL-1 or 1 on KACs and precursor epithelial cells. KACs express IL-1 or 1 at much higher levels than normal AT1 or AT2 cells, making them uniquely sensitive to IL-1-driven inflammation. Panels 4E and 4F project these signals onto the tissue. IL-1 beta maps to macrophage-rich pockets, and IL-1R1 maps to precursor epithelium and reactive pneumocytes. These two compartments sit directly next to each other, forming epithelial pro-inflammatory niches at the earliest lesions. Panel 4G brings together epithelial metaprograms, macrophage programs, and IL-1 beta expression. RP2, the in situ counterparts of KACs, consistently appear in areas enriched with IL-1 beta high macrophages, confirming that these transitional epithelial states develop inside inflammatory microenvironments. Overall, Figure 4 establishes the core idea that early alveolar progenitors aren't simply drifting towards tumour identity. They're being pushed by pro-inflammatory macrophages through IL-1 beta, IL-1R1 signaling. Figure 5 then goes deeper, showing at single-cell spatial resolution how these niches organize themselves around precursor lesions. And here it is. Figure 5 takes everything we saw in Figure 4 and resolves it down to single-cell spatial precision using the Xenium platform. Panels 5A to 5C show that at cellular resolution, the same transitional alveolar population appears, those KACs, along with an inflamed AT2 subset that expresses NF-kappa B related genes. KACs again score highest for the transitional program. Then panels 5D and 5E quantify neighborhoods. KACs, precursor lesions, and inflamed AT2 cells all sit within microenvironments enriched with IL-1 beta high macrophages. Panel 5F visualizes the key interaction. Precursor epithelial cells and reactive pneumocytes express IL-1R1, and they sit immediately adjacent to macrophages expressing IL-1 beta. Panels 5G through 5L validate this in an independent cohort. Across dozens of lesions, precursor regions consistently show more IL-1 beta high macrophages, more IL-1R1 positive epithelial cells, and tighter epithelial immune neighborhoods than invasive LUAD. Figure 5 confirms at true single-cell resolution that early LUAD precursors are defined by tightly organized epithelial pro-inflammatory niches centered on IL-1 beta producing macrophages and IL-1R1 positive alveolar intermediates. Figure 6 moves into functional models, showing that the same epithelial pro-inflammatory niches seen in humans also drive early tumor initiation in a KRAS mutant mouse carcinogenesis system. That's right, they even did mouse work. Panels 6A to 6C show that in NNK-exposed mice, KACs again sit between AT1, AT2, and tumor cells, and they upregulate inflammatory and NF-kappa B associated pathways more strongly than any other alveolar subset. Panel 6D confirms that mouse KACs express the IL-1 receptor, IL-1R1, at the highest levels across the epithelial lineage. This mirrors the human data and sets up the signaling axis mechanistically. Then panels 6E and 6F map KAC signatures and inflammatory signatures onto mouse lung tissue. Even before visible tumours form, early lesions are surrounded by macrophage-rich IL-1 beta high niches, with KACs clustering at that interface. Panel 6G shows dense IL-1 beta IL-1R1 signaling around emerging lesions. Signals originate in macrophages and flow directly into KAC-rich epithelial regions. Panels 6H through 6K translate this into a functional readout. KC-rich organoids, yes, they did organoids too, derived from AT2 cells, expand more when exposed to IL-1 beta. Co-culture with lung macrophages has the same effect, increasing both organoid number and size. This shows that IL-1 beta is not just correlated with KC biology, it actively promotes progenitor expansion. Figure 6 demonstrates that IL-1 beta-driven inflammatory niches are not just spatially associated with early LUAD, they cause alveolar progenitors to expand and transition towards tumour identity. So here's figure 7. It asks the key translational question: if IL-1 beta-driven inflammatory niches promote early tumour development, can blocking the signal actually intercept disease? Panels 7a to 7c show that treating mice with an anti-IL-1 beta antibody significantly reduces the number of transitional KAC-like epithelial cells after carcinogen exposure. These mice show fewer early lesions and less epithelial plasticity overall. Panels 7D and 7E demonstrate that IL-1 beta blockade also dampens the local inflammatory environment, lowering expression of cytokines and myeloid activation markers that normally support the niche around KACs. Panel 7F illustrates the importance of timing. IL-1 beta blockade during the precancerous window reduces tumour burden, but starting the therapy later, once the lesions have progressed, has little effect. Panel 7G and 7H show that combining IL-1 beta inhibition with anti-PD-1 therapy produces an even stronger reduction in tumour development. The combination lowers both epithelial transitions and pro-tumour immune activation. Overall, figure 7 demonstrates that IL-1 beta is not just a marker of early LUAD niches, it's a functional driver. Blocking IL-1 beta can intercept early carcinogenesis, especially when given before lesions become invasive, and it synergizes with checkpoint blockade. This completes the paper's model. Alveolar progenitors, shaped by inflammatory IL-1b niches, form the earliest precancer footholds of LUAD, and those niches are, critically, targetable. Okay, before we go any further, spatial transcriptomics is a really complicated technique. And if you've made it through the panel by panel, understanding the lung side, but not getting the technical side, I feel you. Let's do a quick spatial transcriptomics 101. If it's not clicked so far, this is essentially a mix between transcriptomics and imaging, like IHC or immunofluorescence. So with transcriptomics, where you get sort of PCR, but on a huge scale with thousands of genes, and it's all numbers, here it's not just numbers, it's images too. So you're looking at gene expression. Gene X is high, gene Y is low, and so forth. But not just as an average across your whole sample. You see, normally with transcriptomics, you take a piece of lung, dissolve it, digest it down, and look at all those genes on average across the whole blob of goo. Whereas in spatial transcriptomics, you don't. You keep the tissue intact, and it allows you to see where that gene expression is across your slide, just like how you would with immunohistochemistry or immunofluorescence. So rather than looking at protein, you're looking at genes, but you're still getting that spatial map across the tissue. Now, there's a few different spatial transcriptomics technologies available, and these authors actually do use a few of them. Some of them are in situ, some are not. But either way, you're getting gene expression and you're getting it across a piece of tissue. It might be hundreds of genes, thousands of genes, it might even be a whole transcriptome, but you're seeing it spatially. That's why this technique is so exciting. Now, it's been around for a few years. It's not quite brand new anymore. And the data analysis side has gotten really advanced. It's now allowing biologists to actually answer bigger questions and delve deeper into the data. So you can see things like epithelial states, inflammatory niches, clonal patterns. You can look at different cell types. You can really do a lot with this type of data. And these authors certainly have. It's a fantastic resource. It's a really, really nicely done piece of work. And I know I've said it a few times, but I'll say it again. It looks incredibly expensive. Okay, so back to the big picture findings of this study. If you're studying lung cancer for maybe your dissertation or your PhD or postdoc, then you're going to want to know the big take homes for lung cancer research. And there certainly are some here. You know yourself that a lot of papers just offer some incremental developments in the field. This is not one of them. This offers a few really big picture shifts in how we understand the earliest steps in tumour development. It reframes lung cancer not just as a genetic disease, but as a spatially organized, niche-driven process, something that emerges from the interaction between epithelial progenitors, inflammatory cells, and the tissue architecture that surrounds them. So there's a few high-level concepts to take away. Firstly, early LUAD is a story of cell state transitions, not just mutations. Traditional lung cancer models often focus on KRAS or EGFR mutations, copy number alterations, clonal evolution. Those remain critical, but this paper shows that cellular reprogramming precedes visible cancer. So the key insight is that LUAD doesn't begin with a fully transformed tumour cell. It starts with transitional alveolar cells, the KRT8 high alveolar intermediates, as they call them here, KACs. These cells sit between AT2 cells, the presumed cell of origin, and tumour epithelial cells. They aren't fully malignant, but they've already lost some alveolar identity and gained early tumour-like programs. Why this matters for your dissertation is that it shifts the question from which mutation starts LUAD to which cell states make the lung epithelial vulnerable to transformation. The second thing to take away is that LUAD precursors develop inside pro-inflammatory niches. One of the biggest conceptual advances from spatial biology is this. Early LUAD is not just an epithelial problem, it's a niche problem. The paper shows that early epithelial transitions happen next to IL-1 beta producing macrophages, forming tightly organized micro-niches. These niches appear early, even in AAH or AIS. They concentrate IL-1 beta, they contain KACs and inflamed AT2 cells, and are largely absent in normal lung tissue. This means early lung cancer is shaped not only by intrinsic genetic changes, but also by local immune-driven microenvironments. For dissertations, you should note that this ties into wider themes in tumour immunology and microenvironment research. LUAD development requires both epithelial plasticity and inflammatory signalling. Thirdly, clonal evolution in LUAD is spatially patterned. Using spatially inferred copy number alterations, the paper identifies 3 evolutionary patterns across patients. Some precursor lesions feed directly into LUAD, some diverge early, some appear evolutionarily independent. The key insight is that precancerous lesions are heterogeneous. Not all will progress, and not all progress along the same trajectory. And importantly, early clones often map to reactive pneumocytes, not to morphologically obvious tumour cells. For dissertations, this highlights why sampling bias is such a problem in lung cancer. Tiny areas of transitional epithelium may contain the earliest malignant clones. Fourthly, transitional alveolar cells, KACs, are spatially anchored and biologically conserved. KACs aren't random intermediates. Across this atlas, they cluster at lesion borders, co-localize with IL-1 beta macrophages, express high IL-1R1, carry early clonal alterations, show conserved transitional programs found in other cancers. They represent a stable, reproducible biological state. For dissertations, this suggests that transitional progenitor states might be a universal feature of epithelial cancers, not just LUAD. Fifthly, early intervention might be possible, but only in a precancer window. One of the most important translational findings here is that blocking IL-1 beta prevents tumour initiation, but only when given early. Once lesions are more established, so MIA or LUAD, IL-1 beta blockade loses efficacy. The niches are less dominant. The tumour is less dependent on the IL-1 beta program. But when combined with PD-1 blockade and early disease, the effect is stronger. For dissertations, this ties into prevention biology and chemointervention. LUAD may have a true window of vulnerability before invasion. This brings about questions around detection. Now, sixth, a big conceptual shift is that LUAD emerges from co-evolution of epithelial and immune niches. The major contribution of this paper is this integrated model. 1. AT2 cells undergo stress and transition into KACs. 2. Macrophages create IL-1 beta inflammatory niches. 3. KACs express IL-1R1 and respond to this inflammation. 4. Early clones expand within these niches. 5. Spatial structure stabilizes these early programs. 6. Some precursors progress, others don't. 7. Interception is possible if the niche epithelial interaction is disrupted early. This is a co-evolution model, not a linear mutation model. For dissertations, this aligns with modern views of cancer as an ecosystem rather than a purely genetic disease. Okay, so how can you use these findings in your dissertation? There's a few different angles you can leverage. You can discuss attitude, KAC to tumour transition as a continuum, and so frame it as the role of transitional epithelial states in early LUAD. You can use IL-1 beta, IL-1R1 as a case study, so microenvironmental niches in lung carcinogenesis. You can talk about spatial transcriptomics in lung cancer research more broadly as a methodology, how resolving expression in place reveals biology hidden in single-cell data. Or you could take the angle that clonal evolution in pre-invasive lesions is the point here, so discuss the heterogeneity of precursor lesion trajectories. Or if you want to focus more on a treatment story, you could say that targetable vulnerabilities in early lung cancer are identified here and kind of emphasize that pre-cancer intervention window, which seems to be really important from their data. Okay, that's it for this video discussing this spatial map of early LUAD. If you want to watch more videos like this, you can do so at my channel. I talk about recent, high-quality, high-impact papers, usually a little bit more concisely than this one. If you prefer a weekly digest format with either a quick list of the top 5 papers I'm reading this week or maybe a deep dive on one of them in written format, then you can see that too. The link is in the description to that. It's up to you. Happy reading and enjoy.